it looks like zoology, but in this case, it's very applied zoology, because the biology of the tick really tightly connects to the epidemiology of the diseases they do transmit. And if we want to understand the epidemiology, or if we want to control the tick-borne diseases in animals, we want to understand the epidemiology. And if you want to understand the epidemiology, we must be sure that we truly know the basic biological things which are behind. And I try to put all these things together into my talk. You will see if I manage that or not. But anyway, let's start. Oh, it doesn't work. Wait. Yep, it does. So everyone knows, and I already mentioned that the ticks are ectoparasitic arthropods. And in fact, these are highly specialized ectoparasitic mites. So they are relatively closely related to other groups of mites. And you know that they belong to all the metastigmata. So all metastigmata can be called ticks in English. So let's look to the very basic things. I apologize if you find them too basic, but I still think that it's good to really review the basic features. So what's important is that they are obligatory hematophagous. So these arthropods are able to feed only on blood of animals. So it's the really high specialization among arthropods as such. They are parasitic in all groups of terrestrial vertebrates. And of course, they primarily avoid aquatic vertebrates, but still we find ticks on some semi-aquatic vertebrates, like, for example, snakes, frogs, marine mammals, or not marine, but semi-marine mammals. So all this can be affected by ticks. We can say, or in every textbook you read, that the ticks have global distribution, which says a lot, but in the same time it says nothing, because there are huge local differences. So there is nothing like global tick, or maybe one, but even that is not very global. So, and this is what's behind all the tick-borne diseases, that there are huge local differences between the continents, there are huge differences between the countries, but there are also huge differences even between the provinces in individual countries. So I mean, the differences in tick fauna, and then logically, this is reflected in the differences in tick-borne diseases. So I'm from the Czech Republic, so the situation with tick-borne diseases in my country is absolutely totally different from southern Italy. Why? Because we simply have different tick species, and they do have different ecology. If you look at the pathology of ticks, or their medical and veterinary importance, we clearly see two lineages. Either the ticks are directly or causing the direct harm to their hosts. Doesn't matter if these are animals or humans. Of course, they are annoying, they are blood feeding, they are causing tissue damage, they can cause intoxication. We'll go a bit more into that. So this is the direct impact, but the major importance of them is as vectors of the pathogen. This can be bacterial, viruses, protohelminths, and they are causing tick-borne diseases, or we can say also tick-associated diseases, but tick-borne is nowadays broadly used term. Yeah. And because the ticks are, of course, well known, and because they are so tightly associated with their hosts, and also the pathogens they transmit are really closely associated with particular tick species, the ticks were the first arthropods that were, during the previous centuries, demonstrated to be vectors of diseases. It was specifically the babesiosis. We will speak about it next week. And it really started almost a race for discoveries of different pathogens transmitted by different arthropods, like malaria, leishmania, and all these trypanosomes. But this historical story, or this historical journey, started with ticks and babesiosis. So that was the first vector-borne disease described as being transmitted by parasitic arthropods. Yeah. Logically, both because of direct impact on the hosts and also because of transmission and diseases, leads to really huge economic impact of ticks on the agriculture, animal husbandry, and also human societies through the diseases, treatment, prophylaxis, and so on and so on. So it's really important group of pathogens or group of vectors, probably second to mosquitoes. If you look more to human medicine or vector-borne diseases of humans, then mosquitoes are in general considered being more important vectors. Why? Because of malaria, of course. It's still dengue, for example, and West Nile fever. So those protozoa or viral infections that are transmitted by mosquitoes, they are really among the most important infectious diseases of humankind. If we go more to the animals, then of course, even in animals, we see the diseases that are transmitted by mosquitoes, but definitely the area of vector-borne diseases of animals is dominated by the diseases that are transmitted by ticks, specifically if we look at herbivores. So if you look at to the ticks as a group of organisms, it's, to be honest, not big. It includes less than 1,000. They say it's about 900 different species, which are divided into 3 families. It can sound as a huge diversity, 900, but you know that in general, the arthropods, they are the most diversified organisms on multicellular organisms on Earth. So compared to the overall diversity of arthropods, ticks are nothing. 900 species, it's nothing. If we could compare to dipterans, to beetles, to butterflies, and all these, 900, it's nothing. If you look to these families that ticks divide to, you possibly might know that there are 2 major families, Ixodidae, which we call hard ticks, and Argacidae, which are called soft ticks. Today, I will speak mainly about the Ixodidae, about the hard ticks. Yeah. Also, the importance of these families is also reflected in the diversity, or vice versa. So there are more than 700 species of hard ticks and only less than 200 species of soft ticks. And there is one very particular species of the family Nuttallelidae, which is biologically interesting, but not really important. So we don't really need to speak about it. And in general, we can say, and it's in general pattern of the biodiversity, that the diversity of ticks is higher in the tropics. And there are only few species that are able to persist or to penetrate into deep polar regions, specifically in the north. And as I said before, the different areas of the world, they have different diversity and also different composition of the tick fauna. So very briefly, just morphological insight into the soft ticks, into family Argacidae, which are less known than the hard ticks. They are called soft ticks because their body is lacking the chitinous scutum on the surface. So their body looks like a bit covered with a skin. This is one of the important features. The second important feature is that if you look at these ticks from the dorsal side, basically we don't see the mouth part, we don't see the basis of the legs and so on. Everything is hidden on the ventral surface. Of course, if you flip the ticks, then you see the so-called head or capitulum, which is located ventrally. I don't know if you see it somewhere here. So on this picture, we see one of the most common soft ticks, it's Argas. In general, if you look at these 2 genera, the ticks of the genus Ornithodoros are more associated with mammals, even though some of them can be parasites of reptiles and so on. The genus Argas is primarily associated with birds. So here you see the same on the color picture, Argas reflexus, relatively common European species that is associated both with poultry as well as with free-ranging birds. You can find colonies of Argas even in the cities where they live together with pigeons. The ticks of the family Argacidae, they have quite different biology. The soft ticks, they differ a lot in their biology from the hard ticks.
Sorry.
Yes.
Can I ask you why they called soft? They are called soft.
Yeah, because they lack the dorsal shield, which is in hard ticks. I will show you a picture soon. In hard ticks, the dorsal part is covered by so-called scutum. Yeah, which is highly chitinous, very hard plate, which is covering the body. And this is missing in Argacidae. So this is why we call them soft ticks. And they are not that much soft, but they are definitely softer than the hard ticks. And because of this, they can also extend the volume of their bodies quite significantly, which is typical in hard ticks. It's typical for females, but in soft ticks, both males and females can really enlarge and get full of their bodies. The advantage of this, of the fact that they are really flattened, is associated with their biology, because they don't attach permanently on their host or for long period of time on their host. And this is big difference from the hard ticks, from the Ixodidae. In the Ixodidae, you know it from yourself, you know it from your dogs, cats, and so on. The tick becomes attached and persists attached to the host skin for at least a couple of days. In Argacidae, it's really different. They in this group of ticks, only the larvae attaches for a longer period of time, but the adults, they are only coming to feed at night on their hosts for a very short period of time. And for the rest of the day, they are hidden somewhere. They are hidden in the crevices of soil, they are hidden in the crevices of walls, they are hidden inside the bird nests, and they are simply waiting for their host to come. And then at night, they are really actively moving. They crawl out from their shelters, and they suck the blood for just maximum couple of minutes, usually even faster. So they are able to engorge very fast. In the next picture, you see them hidden in the crevice. You see how flat they are when they are unfed. And this is how they can survive really long period of time. Normally, they can survive months and months without any access to food. And the extremes that were demonstrated experimentally are more than 10 years. So this soft little creature is able to wait somewhere 10 years for the host to come and to feed it with its blood. So it's quite special or unique biology. And this is why normally we don't find them on the animals. You might live in the area where, for example, Argas is really common in poultry, and you simply don't see it unless you go to the stables or poultry houses and you really intensively look for them, because they are always hidden. And the same is true for Ornithodoros, which is parasitizing mainly mammals. These ticks are also important vectors. They are important vectors of some species of Borrelia, and they are also important vectors of some viruses. Do you know any virus which is now very popular in Europe, which is transmitted by soft ticks originally in Africa?
No.
You must know. What is the virus that people in Italy talk most in the past few months? Mostly discussed animal virus in Italy.
Coronavirus.
Not really. Good, almost there, but not really. Not during this summer. Coronavirus is over, and it's not transmitted by ticks. But there is one viral disease which everyone speaks about in Italy.
Peste aviaria.
It's associated with different animals. Associated with pigs.
Rabbia.
No, rabbia, no.
Rogna.
It's a disease which is really threatening prosciutto industry.
La peste suina, peste.
Yeah. African swine fever. Yeah. ASF, African swine fever, is a virus which is primarily transmitted in Africa. It's transmitted by ornithodoros ticks in the wild bush pigs or warthogs, the free-ranging, free-living African suids, African pigs. However, in domestic pigs, it can be, because the virus survives a lot in the environment. So in European situation, it is transmitted mainly by direct contact of the animals or through contaminated environment. But originally in Africa, it's typical vector-borne disease transmitted by ornithodoros, by the soft ticks. But anyway, let's move to the Ixodidae, to the true ticks, to hard ticks. A little bit about their diversity and phylogeny, but as I said before, I'm not really going to dive deep into this because it's not a field for veterinarians, in my opinion. So, as we said before, this is the most diversified group of ticks. The major differences from the Argacidae are in morphology. They have the dorsal sheath, as you see here on these ticks. This is hyaloma, and you see this is male, and you see that the whole dorsal part of the body, upper part of the body, is covered by scutum or dorsal sheath, which is a very solid plate that protects the tick from the dorsal side. So this is one important thing. Also, you see that the cranial part is visible from the dorsal view. So we clearly see the hypostome and the palps in this tick. So this is the morphology, much longer legs as well, and so on. And the biology, it's in the number of the blood meals. As we discussed before, the soft ticks, they suck blood repeatedly, only for a short period of time. In hard ticks, it's very different. Each stage of a hard tick sucks blood only once. And you know that there are 3 stages in the life cycle. There is larva, there is nymph, and there is adult. And each of these stages sucks the blood only once. So it's not very enjoyable lifestyle. Just imagine that you have just 3 meals in all your life. It would be difficult to decide what to take. Pizza, pasta, all this nice type of seafood and so on. But ticks, they have it easy. They feed only on blood. So 3 blood meals in their whole life. So it's more than 700 species that are divided into 14 genera. Some of these genera are more important than the others. So here I mentioned only those that are really considered important. And by taxonomies, they are divided into 2 groups, so-called prostriata, based on the position of the anterior anal groove, which is a morphological feature on the ventral part of the body. And this group includes the genus Ixodes, which is the largest, the most numerous genus of the ticks, including also several species in Europe. And the second group is Metastriata, having so-called posterior anal groove, and it includes a range of the genera, but the most important from the medical and veterinary perspective is Dermacentor, Hemaphysalis, Rhipicephalus, Hyaloma, and Amblyoma. So those are the tick of importance. The others are taxonomically interesting. They are, for example, on reptiles and so on, but we cannot really say that they represent an important topic or target in veterinary parasitology. So if you look into general morphology, as I said before, there are 3 stages. We can look first to the nail on the picture, to the thumb, and as you see there, and as you see also on the drawing on the very right side, the larvae are very small, and you know it, or maybe you even don't notice their presence on the animals. They are so small that they are difficult to find. That little tiny dot, which is on the thumbnail on the picture, this is larva. Yeah, so it's very difficult to find them in the field, very difficult to find them on the animals because they are little tiny. But still, they are important parasites, and still, in some situations, they can transmit the diseases. We will speak about that later. Then we have nymph, and the larvae, they have 6 legs. It's typical for this group of mites that the larvae, they have only 6 legs. The nymphs and adults, they have 8 legs. You probably know it. Yeah. In the middle of the picture, we see the dorsal and ventral view is the letter C. We see nymph, which is the medium-sized stage you see on the color picture, the one on the right, upper right part of the nail. So this is the nymph of Ixodes ricinus. And then on the left side, we see female and male dorsally and ventrally. A is male, B is female. And here on the dorsal view, which is the upper part of the picture, we see the important difference between male and female, and it's the size of their scutum. We don't go much into morphology, but this is important to know, and this you should really know, how to distinguish males and females. So the B is female, and you see that the scutum, dorsal scutum, which is marked by the letters DS, is covering only part of her body, and the rest it's soft. I would say the rest of the body resembles the soft ticks. While in our male, which is the one on the left side of the picture, you see that the dorsal scutum is covering the whole body of this particular male. So the soft part of body is not visible, covered totally by scutum. It's important, looking at this picture, it's also important to know that some ticks, they have eyes, some genera, some other genera, they do not have. The tick on the picture, on the drawing here is Dermacentor, and it has eyes. So they are visible both on females and the male. I'll try to show you. These are these little tiny things, little tiny spots next to the second pair of legs. These are the eyes. So these ticks, they see quite well. There are other tick genera like Ixodes, which do not have eyes at all. It means they see nothing. Yeah, so they are totally dependent on other senses than the vision. So this is the basic morphology. Yeah. If you look to the ventral side of these ticks, by letters GP is marked the so-called genital pore, which is the opening of the genital organs. If we go to the opposite side of the body, to the caudal side of the body, we see the anal opening. So ticks are among those arthropods where the genital organs are opening to the body surface at a totally different side than the anal opening, which connects to the copulation or mating. I'll show you pictures later how the mating procedure is happening. So this is the very basic morphology. So if we review, larvae, very small, 6 legs, scutum is covering only part of their bodies. Again, the letters DS on the larva. Nymphs in the middle, 8 legs, 4 pair of legs. Again, scutum covering only part of their body. They do not have the genital pore because they are not sexually active. And the adults, females, small scutum, males, large scutum, genital pore or genital opening cranially, anally, caudally is the anal opening or anal pore. So this is the very basic morphology, and I think this is, and the eyes in some genera. So if you look to the cranial part of the body, it's called hypostome, which is the piercing organ that fixing organ using which the ticks are fixed in the skin. Yeah, we see it nicely under the letter C. So this is hypostome with the hooks that fix the tick inside. However, the ticks, it's not fixed only mechanically by these backwards pointing hooks, but also chemically it produces special substances from so-called cement glands, and it helps to fix the tick in the skin. If you look to letter to letter B, which is, it shows quite nicely the palps, or letter B and A. One is dorsal, one is ventral. It shows the palps, which are the organs on side of the hypostome. And it shows also the chelicerae, which are marked by the number C. So the chelicerae, this is the proper organ that cuts the skin. And then the tick inserts the hypostome inside the skin, and the semen glands are starting to produce the chemical substances that fix the tick in skin. And this is why the tick cannot move freely from one side to the another. And this is why every single stage of ticks suck the blood only once. Once the tick is fixed, it remains there and continue sucking the blood for a couple of days. Yeah. This is just the basic key for distinguishing of the major tick genera, which is, in my opinion, enough for veterinarian to distinguish. And we even don't need to go right now through details, or maybe we can briefly, I mean, we can, it's not that complicated. So let's start from the left side. It's typical dichotomic key, yeah? So the first division divides the genus Ixodes and the others based on the anal groove, which is either anterior to the anus or posterior to the anus. Anal groove is this line. Yeah, so you see here it is anterior to the anus. Here it is posterior to the anus. So this divides the major two groups. If we go with those having the posterior groove, four major genera included: Dermacentor, Hemaphysalis, Rhipicephalus, Hyaloma. And in most of the Dermacentor species, they can be distinguished by ornamented dorsal scutum. But just one thing, please, this is only for European ticks. Yeah, this schematic drawing is for ticks in Europe. Because if we go to tropics, there are also other tick genera, like, for example, Aponoma or Amblyoma, which are having nicely ornamented scutum. But if we stay in Europe, ornamented scutum, it immediately means Dermacentor. I show you pictures later. Non-ornamented scutum takes us to another three genera. If the eyes are present, it's Rhipicephalus, Hyaloma. If the eyes are absent, it's Hemaphysalis. And among the Rhipicephalus and Hyaloma, principally we can divide the ticks based on the length of the palpi, but there are also some other differences. And Hyaloma, it's morphologically very specific tick that, especially as adult, can be easily distinguished. In general, we can say that it's relatively easy to distinguish the adult ticks, but it's quite complicated to distinguish the larvae. So for really proper identification of larvae, you need to really take a stereomicroscope or microscope. You need to take some time. You need a key for this, and you should go through the details of morphology to identify the larvae. In the adults, it's much easier. So let's look to biology. So important feature of biology of the tick, we already mentioned some of them, is that they spend, even though they are blood-feeding arthropods, they spend absolute majority of their life outside of the host, which clearly impacts on tick control, for example, or diagnostics and so on. So at any place where the ticks are occurring, only very little proportion, sometimes they say it's less than 1% of the tick population is occurring on the host. The rest is in the environment. So then logically you imagine that even though we control the ticks on the animals, like dogs, we speak about it later, we use collars, we use spot-ons, and so on. We use always how to control the ticks. We are reaching only something like 1% of the population. And this is also why the ticks are so much dependent on the overall ecology of their habitats. If you compare, for example, the ticks and fleas, there's a huge difference because the fleas as adults, they live permanently on the body of their host. So the fleas on the dogs or lice is even better example. The dog lice, they don't care much if the dog is in the forest or if the dog is living in or walking in the city park, if the dog lives in the Czech Republic or Italy, because they live permanently on the host. So the external environment doesn't really impact on their life. In ticks, it's absolutely different. So, as I said before, all stages of ticks are blood feeding. In some species, the males do not feed at all. There are typically 3 stages, we already mentioned that. And there are 3 types of the life cycle based on the frequency of the host changing. The great majority of ticks are so-called 3-host ticks. I'll show you later on some pictures of the life cycles, which means larva, nymph, and adult, they really change the host during this period of life, or in other words, they always detach and live in the environment between the stages. Yeah. Then we have 2-host ticks when the larvae and nymphs are usually living in one animal. And then we have also 1-host tick, in life cycle of which the tick is not really leaving the host. It's molting from larva to nymph and then from nymph to adult, but it's not leaving the host. And this single-host tick or 1-host tick, they evolved typically in association with migratory herbivores. So this is why we see them, for example, in ruminants, originally in Africa or Asia, or we see them also in the Arctic ruminants. Why? Because the hosts, the animals are migrating so huge distances that if the tick falls down from the hosts are simply moving to other place and the tick stays behind without the access to the meal. So this is so they adapted to this migratory lifestyle of their host by single host or 1-host life cycle. Interestingly, the ticks are also usually mating on the host. So males are finding their host. Males are finding females directly on the host, and they can wait for them quite long. And this is why most commonly the male ticks are not attached immediately after they find their host. So, for example, with Dermacentor or Ixodes, if this tick, if a male tick finds a host, he's just waiting somewhere on the body surface for female to come. So, and this is why we see more frequently males crawling over the bodies of the dog, specifically in Dermacentor, than the females. Because while the female finds the host, in this case the dog, it immediately attaches, more or less waiting for male to come. And the strategy of males is different. They are walking or crawling over the bodies of their hosts, looking for the females that are already attached. On the photograph here, we see a female together with male during mating. Mating is very specific, and you also see it on the picture of Ixodes ricinus in the middle. Mating is very specific procedure, and what's specific is that the sperm of males is transported to genital organ of female by the hypostome. Yeah, so the male first sucks. It's quite bizarre, to be honest. Yeah, the male, quite bizarre form of oral sex, we can say. The male sucks his own sperm into the hypostome and then transmit that to the genital pore of a female, inserting the hypostome inside the female's genital pore. This is why the typical copulation, this is in the picture in the middle, or the ticks in the typical position for copulating ticks is like that. Yeah. The male is sitting on a kind of caudally on the ventral part of the female's body. So it's very unusual, not that unusual. It's not the only arthropode that is mating this way. Spiders, they are doing it also quite similarly, some, but for ticks, it's very characteristic. And if we continue in the life cycle, so this is the mating, then female continues sucking the blood, extending her body, slowly becoming engorged. And when she is fully engorged, like this Ixodidae ricinus female, she's simply detaching herself, falling into the environment, hiding herself somewhere, and waiting until the development of the eggs is finished. And then she starts laying the eggs. I'll try to show you the video on the next picture, which is an adult female of Rhipicephalus sanguineus, the brown dog tick. She just fell down from the host, and now she's looking where to hide to wait for the eggs to be developed. So this is the really largest stage in the tick life cycle. You for sure know them. So they are quite actively moving. Yeah. That's it. She doesn't survive long on the sun-exposed surfaces, so she really tries hard to hide somewhere. And this is the final period of life of tick female. She's laying the eggs. Somewhere in the soil, we can say, or hidden somewhere under the stone. For example, hyaloma, they commonly hide under stones on the pastures and lay the eggs there. Because the eggs, they need humidity. They should be protected from sun. And it takes a couple of weeks for the eggs to develop, and then the larvae are hatching. What's important that some of the pathogens, we speak about it later today, but mainly in next week, some pathogens are transmitted transovarially. So the female is infected on the previous host. She lays the eggs which are infected specifically by viruses. And each of the larvae, or theoretically each of larvae that hatches from clutch, is infected by the virus and spreads the infection further. So this is the egg laying. And this is the larva. They are almost microscopic. This is Ixodidae ricinus. Larvae of Ixodidae are really, really tiny. So you really need a microscope to identify them and to see them properly. And you see what we mentioned before, 3 pairs of legs. Typical feature for larva. Okay. This is just schematic drawing of the 2 and 1 host life cycle of the tick. Most of the ticks, they have 3 host life cycle. We go to that later. But some of them, they are 2 host life cycle and 1 host life cycle. We already characterize that. If we start from the left, some Rhipicephalus from herbivores, Rhipicephalus microplus typically, or Rhipicephalus annulatus, which are the most important ticks on cattle. They are only single host ticks. So as you see there, 3A, 3B, 3C are the ticks remaining on the host. And then the engorged gravid female lays the eggs in the environment. Yeah, so she is number 4. She drops off the host, lay the egg in the environment. Larvae are hatching. They have 6 legs, as you see on the drawing. and they are attaching to another cow, and the rest of the life cycle is on that. It has a really huge practical implication. We'll speak about it later, but I can tell you even now that these are the only ticks that we can really target by the vaccination. Why? Because if we have a pasture, closed pasture with a herd of cattle, we can vaccinate all the animals, and all the tick stages which are actively living or sucking the blood sucks the blood on this particular herd of cattle. So we can really eradicate, by vaccination, we can eradicate Rhipicephalus microplus from cattle herds. And the same way also by treatment, Rhipicephalus annulatus was eradicated in most of the United States, or basically in whole United States in the past century, because all stages live on domestic animals. If you go to ticks with more complicated life cycle, this is virtually really impossible, because even though you vaccinate, for example, every cow, or let's imagine that we develop vaccine for the dogs. So even though if we vaccinate the dogs, the absolute majority of ticks, they are living all around on squirrels, hedgehog, birds, cats, hare, rodents, and so on. And they are happily living regardless we vaccinate or non-vaccinate the animals. And the same is true for the treatment. Yeah, so we treat the dogs, we keep treating them, but absolute majority of the ticks, they are uncontrolled by this way. So Rhipicephalus evertsi, Rhipicephalus bursa, and Hyaloma marginatum are the second group of ticks, which are so-called two-host ticks. And you see, that's logical. If we follow the life cycle, there is nothing special. The larvae, they hatch in the environment. They attach to, for example, to lagomorphs. They suck the blood, then they molt to nymphs. Also the nymphs, they suck the blood on these hosts, and then they molt in the environment, and the adults the next spring are present on cattle. Yeah. Which also, this picture also shows the seasonality of the life cycle and the division of the life cycle into 2 years, at least in temperate zones of Europe and Americas. Yeah, so the first year we have the adults, the second year, and the larvae, and the second year we have the larvae and nymphs on the hosts. So typical long duration of the two-host life cycle is 2 years. Later we discuss the three-host life cycle of Ixodes ricinus. If we speak about the ticks and the environment, it's logical that ticks, they should have a strategy how to find their host. Otherwise, they sooner or later die by starving in the environment if they really cannot locate the host and actively attach to them by some way. There are basically 2 or 3 types, we can say 3 types of the host finding strategy among the ticks. There are so-called questing ticks, which is, for example, Dermacentor on the picture, it's Dermacentor marginatus. And the questing means that the ticks are sitting and waiting for the host to come. So usually they crawl to the top part of the vegetation, to the top of the grass or to the plants, and they sit and wait. They stretch their first legs because they have the glands, I would say the sniffing glands, the glands by which they absorb the chemical stimuli from the air are on the front legs. So they stretch the front legs, somehow moving with them, waving with the front legs, as this tick is showing, waiting for the host to come. When the host is approaching, the tick more or less, we cannot say jumps, but simply attached to the host. When the dog is passing through the grass, the tick attaches to that. Then we have second extreme so this is quite extreme way, and only some species of the ticks, they show this behavior, typically Ixodes ricinus or Dermacentor. Then we have so-called hunting ticks, which are actively searching for their host. The typical hunting tick is Hyaloma. They are really crawling long distances, we can say hundreds of meters, to find a host. They have the eyes, so they have quite good vision, and they are crawling around, and when they see the animal, simply they crawl on that and they attach. And then kind of mixture of previous two, not very differentiated strategy is so-called cohabitation, which means the ticks are somehow living together with the hosts. I would say the typical of these ticks is Rhipicephalus. So they live in relatively high densities around the animals. So when there is an opportunity, they simply crawl on the animal and get attached. If you look to the host spectrum of the ticks, not surprisingly, we find the ticks which have very broad host spectrum, typically Ixodes ricinus. We have the ticks which have quite narrow host spectrum, and we have the ticks which are very specific. If we go to the specific ticks in Mediterranean, we can mention, for example, Hyaloma aegyptium. The tick which is as adult feeding the blood only on land tortoises. Because we already know that the ticks get attached, detached from their hosts in individual stages, we already know that the transmission of the diseases is connected to that. Then it's obvious that the host spectrum or host specificity of the ticks is very tightly connected with the transmission of tick-borne diseases or with the epidemiology of tick-borne diseases. Yeah. So if the tick is very specific, like Boophilus or Rhipicephalus microplus for cattle, then logically such a tick species is a dominant or the only vector for several tick-borne diseases of cattle, for example, for Babesia. Yeah. If we take the second extreme, I'll go to that later, but we can discuss it now, like Ixodes ricinus, it's absolutely generalistic tick. And most of the pathogens which are associated with these ticks are also generalists, typically Borrelia burgdorferi or Anaplasma phagocytophilum. So they can more or less infect any type of mammalian host, and they cycle in quite complex life cycles or networks in the ecosystems. We discussed that specifically in Ixodes ricinus a bit later. So let's look to the tick distribution, because tick distribution is something that we already discussed in the beginning of the talk, which stays behind the geographic differences and ecological differences in the occurrence of tick-borne diseases. So as a background for this, or background information for this, before we go deeply into this map, I will show the same map later in bigger format, so no worries. Here it is only just to see different colors. Different colors on the map are different ticks. So it's obvious that even in Europe, the tick fauna is really differing between Mediterranean region, between Central Europe, and between the North. Yeah. So we already discussed in the beginning that ticks are globally distributed. Yes, they are. They are almost everywhere. But if you look to individual species, we have to say no. There is nothing like globally distributed tick. Each tick is living in some very particular geographic area, and also each tick is occupying very particular ecological niche. It's interesting biologically, but it has enormous importance in epidemiology of the transmitted diseases. Yeah. Logically, the ticks that are having the strict host specificity, they are so-called co-distributed with their host. It's not surprising, but it's good to keep it in mind. If we have Hyaloma that is associated with tortoises, then logically this tick cannot be found in the areas where the tortoises are not occurring. I think it's obvious. Interestingly, some species are considered invasive, which means they are relatively fast or speedily extending the areas of their occurrence, typically Rhipicephalus microplus, Rhipicephalus sanguineus, or Dermacentor reticulatus. I'll show you examples a bit later. Oh no, I'll show examples right now. So when I show example of in my talk of two invasive tick species, but one in particular, because this is the tick we work with in the Czech Republic. It's important vector of the diseases, and it's Dermacentor reticulatus. If you look to this map, Dermacentor reticulatus is the tick in the circle on the right side. It's not really clearly visible because it's orange dots. Or it's marked by orange dots. So it's somehow in the middle of Europe, more or less north of Alps. It's living also in northern Italy, so it's not only north of Alps, but most of the population is in the north of Alps. While the other tick species which are on this picture, it's Ixodes ricinus, which is the uppermost tick, which is the dark red. They are living in Central and Northern Europe. And by green color, the green colors is here for Epicephalus sanguineus ticks, which are living in the Mediterranean, as you see here, including Sardinia, for example. Yeah, but let's go to Dermacentor reticulatus, the tick which is here in the middle. It's a tick which is very easy to distinguish morphologically, very easy to identify, because it has that ornamented scutum. And on these two pictures, you also nicely see the differences between males and females. Which one is female? Oh, it's a question to you.
The right one.
No.
No.
Female male is dorsum covered with scutum. The right one is male. The left one is female. Yeah, because you see on the tick on the left side that the scutum, which is more or less whitish with some dark pattern, is covering only the first third of the body. So this is female. Yeah. Male is on the right. But anyway, because this is what? Again, the same question. This is male or female? If you compare this, yeah, this is male for sure. And these are quite huge ticks, which can be relatively easily found or spotted by people. And this was a topic for our research in the Czech Republic in past years because of two good reasons. First of all, it's the only vector of canine babesiosis caused by Babesia canis, which is an important pathogen of dogs in Europe, throughout Europe. And second, it's considered being invasive species, or at least species with very rapidly changing distribution. Yeah, so we started a cities. Sorry, this is in Czech because it's a website for citizen science campaign when we really asked the people and the dog owners in the Czech Republic to send us either the pictures or the ticks they find on the dogs to monitor the distribution of Epicephalus of Dermacentor reticulatus. As you see, even on these pictures, it's very easy to distinguish between Ixodes, which is on the right side, and Dermacentor, which is on the left side. Dermacentor. And this is quite interesting. The situation in the Czech Republic that we discovered very recently. Just for you to know, because I don't have a historical distribution picture here in the presentation, maybe later. Historically, the tick was known only in the extreme southeast of the country. Where we started to work with Dermacentor reticulatus, it was present only here. I don't know if you see my mouse or cursor. It's only here in the very right lower southeastern corner of the country. Then we asked the people to send us the ticks, and surprisingly, in past 10 years, we received the ticks from all around the Czech Republic. Most of them, these were individual ticks sent by the people, but in some situations, like in the northwest of the country, we already see the ticks are apparently grouping, and then the new populations are emerging in these areas. Yeah. Which is quite important because the canine babesiosis was historically absent in the Czech Republic. And now we have the situation when the tick as a major vector or the only vector of canine babesiosis caused by Babesia canis, we have to stress, is living and distributed all around the country. I think I will go to that a little bit later once again. This is just a show that for the tick distribution, and the same is happening in many other countries around, for the tick distribution, we can use the citizen science, and the vets, of course, are also important part of this chain, because just by collecting the ticks from your patients, by providing the ticks to people that can identify the ticks, we can really improve our knowledge about the tick distribution in particular areas, which is an absolute background for understanding the distribution of the diseases. So what is the mode of the spreading? When we know that the Dermacentor reticulatus is in past 10 to 15 years really spreading all around Europe, it's not only in the Czech Republic, it's spreading all around Central Europe. What is the mode of the spreading, or what's the reason for the spreading? Is that global warming? There were some hypotheses it's global warming. No, it is not, because it was much earlier discovered north of our country than in the Czech Republic. But we can say that the distribution, or the fast changing distribution of this tick in Europe, it's part of the global change, being associated not with the global warming, but with the global change, being associated with traveling of the dogs. You know that nowadays I speak about the Western world, in Europe. It's really popular, or it's not surprising that the owners, they do every travels together with their dogs. You go for weekend, you take your dog. You go for holidays, you take your dog. You go for jogging or biking or cycling or whatever, you take the dog with you. So no surprise that the ticks that were originally quite localized and distributed only in small areas are nowadays so actively spread, not only within the countries, but also internationally. I'll show you the international invasiveness of Rhipicephalus later. And what, for example, in the Czech Republic, probably also connected to this distribution of the tick are the changes in population of wild carnivores. You know that the populations of wild carnivores all around Europe are growing as a part of so-called rewildering process. We like them, we protect them, we want to see them around us, but then logically these animals are creating reservoirs not only for the vectors, but also for the diseases. And we still have to learn how to live with this phenomenon. And interestingly, specifically the areas in the northwest of the Czech Republic, where we see the most Dermacentor ticks nowadays, are the areas of reappearance of the populations of European wolves. And we do also monitoring of pathogens on the wolves, and principally every single wolf that we see is affected by Dermacentor ticks. So again, no surprise that the population, regardless the control of the ticks on the dogs, the populations of these ticks, they survive logically on free living carnivores, the large carnivores like wolves and jackals, which is more or less new in Central Europe. Yeah.
David.
Yes.
Can you make 10 minutes?
Sure. Super. Good even for me. I'll make small coffee for myself, and we continue in around 12:15 or a little bit later. Yeah, 15-minute break.
Okay.
Super.
Just everybody remains in the room.
Ok.
Ok.
See you guys. See you.
Ragazzi, facciamo una pausa. Ok.
Va bene, prof.
sempre ignorato il problema finché non riguardavano me. Avrei dovuto
So can we start?
Yes. Yes.
I'll just close the door a second. Super. So we stopped exactly here. So welcome back. Do you see the PowerPoint just to make sure everything is fine?
Yes.
Oh, super, perfect. So if we try to connect the tick-borne diseases, the importance of the ticks, and geography and ecology, we can conclude that, or if we ask if the geography or ecology of the ticks is more important for the distribution of the diseases, we should say both. In the previous example of Dermacentor reticulatus in Europe, I showed you the more or less the association between canine babesiosis and the particle tick species, which is spreading geographically. In the next examples, I'll also show you that the ecology of the tick as such and the differences in ecology of different ticks can contribute to really different pattern of infectious diseases. And one of the most complicated stories to understand, and we still don't understand how this is happening, to be honest, in Europe, is the distribution of Hepatozone canis, the protozoan pathogen of carnivores in Europe. And how this is connected or how this is not connected to Rhipicephalus sanguineus, so-called brown dog tick. This is anyway very specific tick, so I will go into details later with this tick. But if we look to Hepatozone canis, I'm sure you probably heard about hepatozone and hepatozoonosis as a canine disease all across the Mediterranean. And if you go to any parasitology textbook, older than very recent, you find identical information. Hepatozone canis is transmitted, and hepatozoonosis is disease of dogs. It's transmitted by Rhipicephalus sanguineus, by the brown dog tick. And it's occurring in Mediterranean, or also more south in the tropics. But if we speak about Europe, it's occurring in Mediterranean. That was more or less fine until relatively recently. This yellow line or orange line is showing more or less the northernmost limit of the distribution of Rhipicephalus sanguineus in Europe. And it was believed until really very recently. When I was studying the veterinary medicine, it's not that recent, but still, it's not that long period ago, I learned that Hepatozone canis is a Mediterranean pathogen. However, very recently, it was discovered that, in fact, it's present also in Central Europe. And it was discovered that it has wildlife reservoirs, and it's absolutely common in foxes all around Europe. We did research in the Czech Republic, 30%, 60%, depends on the locality, of foxes are infected. And what's important, we do not have Rhipicephalus sanguineus. So it's obvious that also other tick species are involved in the transmission of Hepatozone canis. But what's really interesting, even though we have so many positive foxes in Central Europe, Germany, Czech Republic, even UK and so on, we don't have the disease in dogs. In all my life, I saw one clinically infected dog in the Czech Republic, even though we have 50% of foxes positive. We published that, and apparently this tick, and mainly this protozoan, or not this tick, this protozoan has two totally different ecological cycles in Europe. In southern Europe, Mediterranean, your island as well, this is cycling mainly from dogs, or between dogs, among dogs. using Rhipicephalus sanguineus, the brown dog tick. In Central Europe, or even the southern part of Northern Europe, it's a parasite of foxes being transmitted by some Ixodidae ticks from foxes, possibly Ixodes hexagonus, possibly Ixodes crenulatus, maybe some other. But because there is almost no connection between these ticks and the dogs, there is no transmission. So the pathogen remains in the fox population. So it's definitely not geographical pattern as was believed before. I mean, in distribution of hepatozoonosis, but it's ecological pattern. So it's all around Europe, but totally different transmission cycles. So we are more and more diving deep into importance of ticks as in veterinary medicine. So let's look at ticks as a direct causes of diseases. It's relatively it's easy clinically, or like to describe or to see it, or during clinical examination. It's quite complex if you look into details of pathogenesis. So definitely what we know, and it's more and more studied, that there is really complicated interplay between the host and the tick and the pathogen during the tick feeding. The immune system of the host reacts quite complicated way to different stimuli from tick saliva, causing also the inflammations locally and so on, and being really involved in the pathogen invasion. Yeah. What's really essential for understanding the differences in immunity mechanisms, for example, between the mosquitoes that we already discussed and the ticks, we should keep in mind that it's the longevity or length of the exposure. If you are bitten by a mosquito, it takes usually a couple of seconds only. And then the mosquito is engorged or fed with blood and flies away. If we are confronted with a tick presence, our immunity system is exposed for a couple of days to the permanent influx of the antigens from saliva of the ticks, which means it reacts more massively and also more complicated way. Also because the tick saliva is very complex mixture of different proteins and other substances that are having also quite complex functions. And most of the pathogens, which are biologically, not mechanically, but biologically transmitted by the ticks, they are somehow involved in this. And then the chemistry, you can say, of whole tick body or tick glands, and the same way the immunity of the hosts all create very complex environment for the transmission, which is quite intensively studied because it has one really important overlap with the I would say clinical parasitology and treatment of the ticks, and its speed of the pathogen transmission. Because why this is important? Because if you look to the products, later we discuss the treatment. If you look at the products that are used against the ticks, nowadays one of the really important parameters or features of new drugs is so-called speed of killing or speed of kill. Because there are two things going against each other. Imagine that you have a dog which is protected by collar or by spot on, and you have a tick which is coming. Of course, the tick is not killed immediately. It takes some significant period of time for tick to be intoxicated enough by the products that are applied either systemically or on the body surface on the dog. So the tick usually is able to attach itself to the host skin and starts feeding, more or less. And then there is transmission of the pathogen happening more or less in the same time. Some pathogens, they can be transmitted almost immediately from the tick saliva to the host organism. But for some other, it takes time. For example, we know that for Babesia or for Borrelia, it takes several hours. Sometimes some experiments say even it can take more than 12 hours. Some say even it can be more than 1 day before the pathogen really finds the way or gets activated in the tick body and finds the way to the host bloodstream. Then logically, if the tick is killed by the toxicity of the product, during this period, the transmission is not happening. And this is what we want. Because in most of the situations when we treat the dogs against the ticks, we target also, or we want to prevent also the transmission of the pathogens. And this can be only proven experimentally, and because it's more and more difficult to do really scientific experiments with these hosts, I really cannot say that we know enough. About some pathogens, we have almost no idea how fast they are transmitted from tick to the dog. For some other, we have some limited experimental evidence. But anyway, it's really one of the topics for the pharma industry to understand this, to understand how fast the tick is being killed by the product, and in the same way, how fast the pathogen is able to penetrate, to invade the host's body. It can, most of the industry is around the dogs, so it's the same for humans, of course, but there are no really specific products for humans. So most of the pharma industry revolves around the dogs in this case and the livestock. So let's look to the symptoms of the tick infestation. That's not that surprising. Each of you probably seen a couple of dogs or other animals infested by ticks. We can say that they are local and they are systemic. Ticks in general are commonly on the host, localized on the areas of the body with fine skin, which can be head, the surroundings of the eyes, axillary region, the ears, scrotum, the other in cattle, inguinal region, typically for perianal region, typically in horses, in cattle, and so on. So logically, the local changes or local symptoms are localized in the places where the ticks are attached, and sometimes we really have to look at them carefully to find them, because the rest of the body of the host is, of course, unaffected. So more numerous the ticks are, severe the changes are, but on the other hand, in Western society, we tend to exaggerate, or to specifically the dog owners, they tend to overreact to the presence of few ticks. If you look at the picture on the left-hand side, it's a dog in Africa photographed during our vaccination campaigns. And we can say this is more or less a normal situation in Africa among the pastoralist dogs. And most of these dogs are clinically healthy. I don't say that ethically this is fine, but it just shows that the normal dog, the healthiest dog, is able to sustain quite significant population of the ticks on its body without really being harmed. Of course, we are not speaking about the pathogens. On the right side, you see the group of hyaloma on the skin of camels. And because the hyaloma are big ticks with really strong mouth part, they can really cause the local granulomas. They can cause skin thickening and severe local reactions, specifically when the animals are permanently exposed to the tick presence. Yeah. So what is the local effect on the site of bite? Of course, it's traumatic, specifically by those ticks with large rostral part, and specifically by those that are penetrating deep into the mucosa, which is typically amblyoma and hyaloma. Amblyoma in tropics, hyaloma in Europe. And then logically, the local reaction is erythema, pruritus, necrosis, granulomas, nodules, local skin hypertrophy in the situations of long term persistence of the ticks and so on. What's interesting, if you look at on ticks at this picture, there are this is some probably cattle in Africa. How many tick species you see? How many different ticks you can find? Don't look at the size, because the two big ones, these are engorged females. We cannot say which tick is this because they are fully engorged. But if you look to other ticks, if you really look closely, I don't know how big screens you have, but here we have these ticks which are very colorful, this dermacentor. No, sorry, it's Rhipicephalus. It's Rhipicephalus pulchellus. So these are these colorful ticks on the right. If we go to the big tick, for example, on the left, this one, it's hyaloma. Also here it's hyaloma. Also here it's hyaloma. And then there are two colorful ticks which are difficult to distinguish by untrained people, but because I know the situation, or three. So there are 1, 2, and 3 here, and it's amblyoma gemma. Yeah, so there are 3 different genera of ticks present on this individual in the probably perianal region. And we should keep in mind that they really differ in biology. They also differ in the pathogens they transmit, and they also differ in the reactions they produce in the skin. Rhipicephalus pulchellus has really fine mouth part, so it's not really causing any significant harm. In contrast, the hyaloma has the longest and strongest mouth part, so it really goes deep and attach for quite long period of time, causing these small granulomas in the skin. Another examples of local effect of the tick attached to the dogs in this case. This is dermacentor. So let's look more to the systemic symptoms. Of course, if the ticks affect the host in higher numbers, they have significant impact on the host health, which is not that obvious in dogs, cats, carnivores, and so on, but it's apparent specifically in the domestic ruminants. And in them, I'll go to that later in the talk, the tick-borne diseases, they cause really significant economical impact because of systemic symptoms. And it's mainly anemia. If the animal is not dying, and it can die, it's anemia, and it's decrease in production in any means. It's decreased lactation. It's the growth of the animal is slowed down, and the reproduction is impacted. We speak about it a little bit later. But then, so this is very unspecific systemic symptoms, not that much surprising. Don't expect much anemia, honestly. Like, you can hardly see like really pronounced anemia in the tick infestation. This is not a typical. But there are very specific things, I would say syndromes related to specific toxicity of the ticks. One of these diseases is called tick sweat or tick sweating sickness, which is a reaction of the herbivores to the specific proteins, specific toxins from saliva of Hyaloma truncatum, occurring in south parts of Africa and in India. In other situations, we commonly see unspecific toxicity, for example, for the cattle breeds from Europe that are imported or kept in tropical areas where they are exposed to high numbers of the ticks. This is not very specific, usually causing severe dermatitis and metabolic disorders. But then we have very specific situations, which we call tick toxicosis or tick paralysis. And it's overreaction of mammalian hosts to some of the components of the saliva of particular tick species. The tick paralysis after contact with a tick was described for more than 50 tick species, but there are few which are really well known for this. And the most prominent example of a tick which is causing the intoxication is Ixodidae holocyclus in Australia. And every veterinarian in the eastern coast of Australia knows the tick paralysis caused by Ixodidae holocyclus. And it's one of the main reasons why the dogs are protected against the ticks in these areas, because this is life-threatening situation, and even a single tick can simply kill the dog through quite acute intoxication. The similar situations are described in USA, caused by two species of Dermacentor andersoni and Dermacentor variabilis, one in the west, one in the east. And in South Africa, specifically in ruminants, severe toxicoses are described, or the tick paralysis are described after contact with Rhipicephalus evertsi or Ixodes rubicundus. Whatever tick is causing the tick paralysis, it's true intoxication caused by neurotoxins that are directly targeting or blocking neuromuscular synapses in the host body. And it's acute situation when the paralysis is happening within a couple of hours after the tick infestation. And I just want to repeat, even by a single tick. We don't have this in Europe, even though we have some tick paralysis described by European tick species. This is rare. And the most prominent is holocyclus in Australia. So if you travel to Australia or if you study the books for Australian vets, the tick paralysis always has a chapter in there because it's significant. The tick is quite uninteresting even. It's small, brownish tick. You see it here on the right-hand side. So nothing specific. The specific is high toxicity of some of the components of its saliva. Yeah. And another interesting phenomenon I just want to mention, because you can find it during your veterinary practice, specifically in wild carnivores, the massive infestation by ticks usually leads to penetration of the tick individuals under the submucosa, where they die and can cause the inflammation. So we just reported that a couple of years ago with our colleagues. And if you really work with the foxes in some regions, you find it really frequently that the ticks are not localized as ectoparasites on the body surface, but they are localized in the subcutaneous tissues. We still don't know the mechanisms, how this is happening. We know that the tick is not surviving in the subcutis. Sometimes it's not even causing inflammation. If you look to the picture A, for example, you see clearly the ticks in subcutaneous tissue, and there is no inflammation around them. So it's kind of peculiar feature. Several tick species are able to do this. We found both Ixodes ricinus and Dermacentor reticulatus in the subcutaneous tissues. So that was for sure important, ticks as direct pathogens, specifically in the tropics, specifically in the livestock. But as we mentioned repeatedly during this talk, the ticks are an important or even major vectors of pathogens. I'm not going to describe that much about individual pathogens because we will have more lectures on this topic next week. But I want to speak about some principles because they are more associated to the biology of the tick than to parasite itself or the pathogen itself. Let's look in general and the arthropods as vectors of the pathogens and the ways they are transmitted. We have typical horizontal transmission between the vector and the vertebrate. This is what everyone is imagining under the term vector-borne disease. Yeah. How the transmission of the inoculation itself is happening, it's quite complex. It can be either biologic or also called mechanical. In case of mechanical transmission, the mouse part of the vector works as a needle, just mechanically transmitting the pathogens. Typical example can be, for example, trypanosomes, Trypanosoma evansi, and the tabanids. Tabanids are very commonly mechanical vectors because they have really prominent, strong mouse part that is really piercing deep into the skin and can transmit mechanically a lot of things. If you look to biological transmission, it's typically salivary. When the saliva are infected, it's typical for the ticks. In some of them, so-called regurgitation is involved, and in some, the larvae can actively migrate. That can be, in tick-borne diseases, case of Cercopidae filaria. When the tick is attached, the larvae probably actively migrate, and so on. But all this is fine. This is horizontal, simply transmitted by vector from one vertebrate to the another. This we can skip. Then what's interesting, we have also transmission between the vectors, which is so-called generational, from mother to the egg. Or we also distinguish so-called transstadial transmission, which is not really a transmission, to be honest, because it's just describing, by this term, we are describing the situation then when the larva is infected, then larva molts to the nymph remains infected. So this is transstadial. And then we also have so-called reproductive, where some pathogens in some vector-borne diseases, not in the tick-borne diseases, can be transmitted from sperm to the egg, but it's more or less exceptional. And biologically, also in the vertebrates, some of the tick-borne diseases and pathogens are transmitted vertically. And what's interesting, that there can be also a transmission between vector and vector. Reproductive, we already discussed, but it can be also, we discuss in part, but also can be transmission from male to female and female to male, and can be also behavioral, which is quite interesting way of transmission by so-called co-feeding. And this is exactly what's happening in the ticks. Let's imagine the picture when several ticks are sucking the blood together. So when one tick is infected, the pathogens in the point of common parasitization, the pathogen can go directly from one tick to the another, or mechanically through the blood somewhere nearby or tissues, but the host is not biologically involved. So it's quite complicated, and different tick-borne diseases are fitting this differently. Yeah. For example, important, something what I forgot to mention when we discussed the inoculation or the transmission, the biologic one, if you go to the last point here, hepatozone is mentioned here because it has very specific way of transmission from the tick to vertebrate. It's not transmitted, so from vertebrate to tick, it's transmitted by blood feeding, but from tick to vertebrate, it's transmitted by ingestion, which shows that the treatment of such a dog against the ticks not necessarily have impact on the fact that whether the dog is ingesting the ticks or not, because even perfectly treated dog having collar or spot on or whatever can simply eat the tick that is crawling on its body. So it's a very specific way of transmission. This we discussed. So, and we are back to our 3 host ticks, and here I want somehow to stress how the 3 years of 3 host life cycle and different hosts are involved in the pathogen circulation. And this is really what we should keep in mind when we are trying to imagine the epidemiology of the tick-borne diseases. So this is typical European situation, Central Europe, Northern Italy, Ixodes ricinus, common vector of many zoonotic pathogens. And when we speak about the zoonoses, when we speak about the tick-borne diseases that are transmitted to humans, we should keep in mind that none of these diseases is a disease primarily associated with humans. All of them are typical zoonoses in most of the situations with wildlife as reservoirs. So none of those important diseases is transmitted from human to human. And the sources of these diseases are clearly seen on this picture. Yeah. And this is how ricinus lives. Yeah. So larva of Ixodes ricinus, in the first year of her life, lives on the small animals, rodents, birds, lizards. Then, when he can get infected by a range of pathogens associated with this, typically different species of Borrelia. Then the second year, the larva overwinter somewhere in the environment, and the second year molts into the nymph, or she molts first and then overwinter, and then the nymph is living on mid-sized animals, squirrels, hedgehogs, rabbits, hares, and so on. Again molts, and then in the third year, the adult tick is occurring in the environment, questing, finding another host, which are the big carnivores, big herbivores. And where the humans are involved in this, in all, the humans can be affected by the adults, the humans can be affected by the nymphs, and humans can be affected by the larvae. But the composition of the pathogens in these different stages depends on the ecology and the life history of each individual tick, and depends on the ecosystem in which the tick is living. Yeah, so in some areas, and this also explains what the distribution, even though the Ixodes ricinus is distributed more or less homogeneously in Central Europe, the pathogens are not distributed homogeneously. And it depends on the ecology on different localities. So there are some localities where the ticks primarily feed on the ruminants, like deer and roe deer. There are some localities where they more feed on the medium-sized animals and so on. And all this is influencing the circulation of the diseases. Yeah. So this is the table. I'm not gonna really to discuss in details. You can go through that later, showing the major tick species, showing their hosts, and showing the major diseases which are transmitted by these ticks, and also the geographic distribution. And there are huge differences. Yeah. Some ticks, for example, are involved, and usually the viruses and the pathogens transmitted are not that much associated with the tick taxonomy or the tick phylogeny, but they are more associated with the tick ecology because of the wild reservoirs. Yeah, if you, for example, if we look to the tick-borne encephalitis virus, which is here abbreviated as TBEV, it's transmitted by Ixodes ricinus, it's transmitted by Dermacentor reticulatus, it's transmitted by Dermacentor marginatus, all happening in the Palearctic. In all cases, the wild animals, like the micromammals, are considered reservoir hosts. If you look to some other ticks, the spectrum of the diseases is different. Specifically, when we go to Hyaloma and Rhipicephalus, usually they transmit different diseases. They transmit more the Babesia, Anaplasma stuff, and then some viruses. Hyaloma, you possibly know, is involved in emerging disease in Europe, Crimean Congo hemorrhagic fever. It's a viral disease, quite significant disease of animals and humans, transmitted chiefly by Hyaloma ticks. So later, as a homework, you can go through these. I don't think I need to speak about the diagnosis of the ticks. It's very easy to find them on the body surface of the animal. Usually they are noticed by the owner. Commonly, the owner is coming with the dog or cat to vet saying, oh, man, my animal has ticks all around, or my animal had a tick, and so on and so on. Some stages, as we discussed, like the larvae, are difficult to observe. Yeah. So now let's look a little bit more on the ecology of the examples of tick-borne diseases and how they are connected with the tick ecology. You are veterinarians in Europe, or you will be veterinarians. So it means most of your work will be with the dogs, anyway, in most of you. So I summarize here a little bit about the ecology of 3 major ticks that are found in European dogs and the diseases they are transmitting. So wherever you are in Europe, or wherever, I would say, publication you read about the tick-borne diseases in Europe, there is no doubt that there are 3 important ticks transmitting the canine diseases: Ixodes ricinus, Dermacento reticulatus, Rhipicephalus sanguineus. Even visually, they are quite different, and they differ a lot in the ecology, and this is why also the diseases differ a lot. If you look to the diseases transmitted by these ticks, there is minimum overlap. Ixodes ricinus transmitting Anaplasma phagocytophilum, Borrelia burgdorferi, Babesia divergens, tick-borne encephalitis. These are the main pathogens, then some others which are not clear. Dermacento reticulatus, in case of canine parasites, Babesia canis, almost only, nothing more, Babesia canis. This is the vector of Babesia canis, we spoke about it. If you look to the brown dog tick, Rhipicephalus sanguineus, on the right side, if it transmits totally different pathogens, it's a major vector, or it's believed to be major vector of Ehrlichia canis, which is highly pathogenic bacterium in dogs. It's a vector of totally different Babesia than Babesia canis. It's vector of Babesia vogeli, which has different clinical symptoms. It has different pathogenicity. It's less pathogenic than the Babesia canis. It's a vector of zoonotic Rickettsia conori and some other Rickettsia, and also Babesia gibsoni. So, and we already know from the maps that these ticks have different distribution in Europe, and we already know that they also have different ecology. And this is why this spectrum of the diseases is not overlapping, and this is why it is so different. So again, if we repeat what we discussed with Ricinus, it's a generalist. So it's transmitting the mainly bacterial parasites or pathogens, but also Babesia divergens and TBE among this broad spectrum of the hosts involving also humans. Very generalistic, very unspecific pathogens. If you look to its distribution all across Europe, but most common tick in Central Europe. So this is why we see so many infections by Borrelia burgdorferi in the Czech Republic, and you don't see almost any in Italy, because simply it's not dominant tick in Italy. For now, it's even not known in Sardinia. It's well-known tick in Corsica, but it's possibly absent or at least unknown in Sardinia. This is one of our research topics. So no surprise, you don't have that many cases of Lyme disease in humans and also in the dogs, as here in Czech Republic or Germany or Austria. If we go to reticulatus, we already discussed, Central Europe, and it has totally different life cycle, how it works. It's good to imagine that because it explains a lot about epidemiology of canine babesiosis caused by Babesia canis. Dermacento reticulatus has very specific life cycle. The larvae and the nymphs, they live on the rodents, and they live in the burrows of the rodents, so normally you don't see them. To be very honest, I never seen larvae of Dermacento reticulatus in the wild. We keep them in laboratory, sure, but I never seen them in my life. They are somewhere hidden. Even in areas where the adults are very common on the dogs, you never see the larvae. And only the adults parasitize on the dogs. And if you imagine how Babesia transmission works, then logically, only the adults can get infected on the dogs, because it's a canine parasite. Babesia canis is a parasite of dogs. And how they transmit it, there is only way how to transmit that, and it's through the eggs, because they die. So if there is no transovarial transmission, there is no transmission at all. So to be transmitted further, Babesia canis absolutely needs the phenomenon of transoviral transmission. So from these eggs, the larvae that are already containing Babesia canis are hatching. Then the next year they spend on the rodents. The year after they spend also on the rodents. And in the third year, they infect another dog or cat. If they are lucky enough, I mean the tick, to find dog and cat. So in other words, if you have a dog that is clinically affected by Babesia canis, you have clinical babesiosis. It means that the dog was infested by the tick, mother of which was infected 3 years ago on some another dog. So it's quite complex. And this is why it's very difficult to understand how the disease is kind of behaving in the dog populations, because there is a huge time gap. And also the same is true for the diseases transmitted by Ixodes ricinus. Imagine that if, and it's well known, the most of the larvae of Ixodes ricinus suck the blood on the small mammals like rodents. We know that, or you know for sure as well, that the population of rodents, they tend to cycle in the nature with several years interval. So usually every third, every fourth years, there is a peak of the rodent population. So if there is a peak of rodent population, logically, more larvae are finding the meal, and they are more successful, which means 3 years later, in the same place where the mice were common, 3 years later, you find a lot of adult ticks. And also the borreliosis cycle like this. So the clinical human borreliosis usually appears in the years after the peak of the rodent population. Because in the year when rodents are peaking, the larvae are getting infected on them. And so the infection should wait at least 1 or 2 years to be transmitted to humans. So there is always a wave of human borreliosis, not always, but commonly, which is 1 or 2 years delayed after the peak of the rodent population. So it's quite tricky. It's really medicine mixed with ecology. And we are just at the beginning of understanding how these things are happening in the wild. Because all these are diseases of wildlife primarily, even though we detect them in humans. So this is just Babesia. So once I'll skip this, I'll keep it for next week. And because you are living in Italy, because you work with dogs, it can be quite useful to look a little bit more into the brown dog tick, the one that you commonly find in Sardinia. My PhD student Sajjad, he just came from Sardinia. He was collecting the tick, and the most dominant tick he found was the brown dog tick. Because it's so, what we know about this tick in Europe, what we can conclude about this tick in Italy and about the diseases it transmits. Why it is called brown dog tick? Because it's brown. Not much morphological features, simply uniformly brown ticks, quite boring, nothing colorful. Brown tick. This is the distribution in Europe, and it's typically Mediterranean. What's interesting that some of these spots that you see, for example, in yellow color here, but with high probability also many of the red ones, which are in Central and Northern Europe, they are localities invaded by the ticks because of the dog movement. Probably also the ticks in the localities, the populations in northern France and Germany are all invasive. So the tourists traveling from Spain, from Italy, and from Balkan are bringing the ticks, and the ticks are establishing populations. Not important now. We speak more about the south of its distribution. So again, how the life cycle is of this tick. It's totally different again. It's three-host tick, but it stays, all stages stays on carnivores. So this is why it is so dominant vector of the pathogens typically associated with carnivores that we already discussed. Anaplasma platys, Babesia vogeli, Ehrlichia canis. These are canine pathogens. They are not zoonotic that much. And even if they are like Rickettsia conori, they are not being transmitted that frequently because the tick stays quite a lot on the dogs. On the other hand, it's well known specifically in Italy that in the areas where the tick is common on the dogs, also the infections in humans are happening more frequently. So if you look to the dog itself, it's, I would say, well adapted to host even quite numerous populations of Rhipicephalus sanguineus. This is a picture from Africa. And again, you see the clinically healthy dog, pastoral dog. I think that was Uganda, this one, with ear virtually full of the ticks. So I don't say this is nice. I don't say the dog is happy because of this, but still this is normal. Yeah. If you look to the host itself, so it's probably the most distributed tick species because also its host, it's the most distributed. Yeah? There are about 1 billion of the dogs on the planet. And in most of the areas where the dogs on the planet are distributed, also Rhipicephalus sanguineus is being distributed. The tick itself, it's brown dog tick. It looks boring, but finally, it's quite interesting because recently it was revealed that in Europe itself, there are at least 3 species of the tick that were so far considered being Rhipicephalus sanguineus. So now we distinguish between so-called Rhipicephalus sanguineus group, sometimes it's called also Sanguineus sensulato, and Rhipicephalus sanguineus sensu stricto. So we already know that in Europe, we have relatively complex genetic situation, and what we believed is one tick are at least 3 different ticks. But we have to say that we know almost nothing about the pathogens or association of the pathogens with this individual tick species. Yeah. So how is Ehrlichia canis, for example, associated with those species living in Eastern Europe, and how Anaplasma platys, what Babesia vogeli, and so on? And it's quite probable that the fact that we have 3 different ticks that were grouped under one name stays behind the fact that also the distribution of the pathogens is kind of mosaic. So we have to reopen all these issues, and basically we have to start from the very beginning, because what's written in the books, it might not be true anymore, because in the time the books were written, Rhipicephalus in Europe was believed to be one species, but now we know that these are 3 species. Maybe they transmit the same pathogens, but maybe not. And in Eastern Europe, it's even more complicated because there is at least 1 more species coming from Ukraine down along the Black Sea coast. So the situation can be even more complex on Balkan. Italy so far seems to be relatively uniform, and all the ticks we sequence from Italy, there are not many, but those we sequence from Italy is Rhipicephalus sanguineus sensu stricto. So it's the brown dog tick.
David, what do you think we can continue next next week on WIS? Or
For sure, I have few slides, so I can easily finish it next week. So you think we are already over the time?
Okay.
Perfect.
Because I don't know if we have to go to lunch.
Okay, super. Anyway, we keep in mind that we should finish the economic impact of the ticks, and then we continue with hepatozone and other things.
Yeah, because I think if we go late, we can't eat. So it's
super. Even for me, honestly, it's good. I am hungry, so let's go for lunch, everyone.
Okay, because we have time next week, so we can continue.
Okay. Super. Yeah, definitely.
So many thanks, and see you next week.
We continue next week. Super.
Okay.
So,
happy Halloween.
Nice day. Yeah. Bye everyone.
Grazie ragazzi, ci vediamo.
I'm sure your meal will be better than mine.
Okay, yes.
You are in Italy. enjoy for me as well. And see you next week. My pleasure.
Thanks. Ciao ragazzi, ci vediamo.
Arrivederci.
Grazie, arrivederci.
Arrivederci. Su YouTube trovate la registrazione, ok? con i sottotitoli. Ciao.